Well, here I am, flying down the side of Whistler, doing about 65 miles an hour on an 80-degree slope. I am frantic to get to the lodge because, well, I'm afraid of being late to AP biology. So I'll see you in a minute. Oh, and here we are. Aerobic cellular respiration. I love this virtual thing. I'm thinking I might take it up full time. I could be in the lodge at Whistler most days during the school year and just video this thing in. Anyway, before we start on the glycolysis and the citric acid cycle, let's remind ourselves of the overview. We talked about glycolysis is the cutting of sugar into pyruvate. We are oxidizing the glucose, reducing the electron carriers called NAD plus to NADH. We get 2 ATPs per glycolysis, and of course it does not take place in the mitochondria. The pyruvates then go to the citric acid cycle where we continue to oxidize the glucose, I should say the pyruvates, while we reduce our electron carriers and we make a couple ATPs. The electron carriers ferry its electrons to the oxidative phosphorylation stage where it meets up with the electron transport chain. Now, notice the ATP below the glycolysis and below the citric acid cycle. It says that they are substrate level phosphorylation. We haven't really discussed that yet, but today I want to just compare that to the oxidative phosphorylation below the, oxidative phosphorylation step. The substrate level phosphorylation will be accomplished by using an enzyme. The enzyme will grab an ADP, adenosine diphosphate, and it will phosphorylate it with an inorganic phosphate, making it ATP. Now, oxidative phosphorylation will generate by far most of our ATP, 90% of it. Only 10% comes from glycolysis and the citric acid cycle through substrate level phosphorylation. So just keep that in mind as we continue through. Okay, glycolysis, we are going to harvest a few ATPs, but by far the most important will be the NADHs that will ferry electrons to the oxidative phosphorylation. Before we start glycolysis, however, we're going to have to invest 2 ATPs. But we'll get 4 ATPs out of it, netting 2 ATPs. Again, the ATPs aren't worth a whole lot. It's the electrons ferried by the NADH that are much more important. So we're going to have an investment stage and then, of course, a payoff stage. Now, I want to remind you that glycolysis takes place in the cytosol, not in the mitochondria. Glycolysis is the only stage that is in and of itself anaerobic. It does not require oxygen. In fact, if you remember back, fermentation and anaerobic cellular respiration also use the stage of glycolysis. So note that. I want to go to a bioflix. It's a cellular respiration. It's several minutes long, but we will be able to visually see all of this taking place before we continue.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Okay, so let's just look at this review of glycolysis. We go in with a glucose, glycolysis. We'll clip it down and we will glean 2 pyruvates from it. We will also get 2 ATPs and 2 NADHs. Now I want to look at this pyruvate a minute. We have a methyl group, we have a carbon and an oxygen, and then up here we have a carbon with 2 oxygens. Now as we start the next phase, I just want you to watch what happens to those atoms in the pyruvate. Okay, concept 9.3, we're going to start in towards the citric acid cycle. We're certainly going to move the pyruvates into the mitochondria, but we have a bit of a, let's call it the pre-citric acid cycle. We have some other things to do to the pyruvates before we actually enter the citric acid cycle. So here's our pyruvate in the cytosol. We're going to move it into the mitochondrial matrix. Notice we have our 2 two membranes here, but we have to transport it with active transport. So we're going to move it into the mitochondrial matrix. Immediately, we're going to clip off this carbon with the two oxygens, and that is a waste byproduct of this part of metabolism. We're going to oxidize the what's left, making an NADH from the electron carrier, NAD+, and that will go to the electron transport chain. Then with the rest of this, now called acetyl, we're going to hook on, attach on, bond on a coenzyme A. Now, if you remember from honors biology, a coenzyme is generally a biological product, an organic molecule. It's usually a vitamin, in fact, that an enzyme may need in order to become able to be activated and do its job. So we will call this now acetyl coenzyme A, and now we're ready to go into the citric acid cycle. Now, we're going to watch for, let me go back, we're going to watch for what we're going to generate from the citric acid cycle. Look down here, we're going to get out per pyruvate, we're going to get out 1 ATP, 3 NADHs, 1 FADH2, which is another type of electron carrier, and then we're going to oxidize the other pyruvate, so we'll double those numbers per glucose. So here we are, here's the pre-citric acid cycle, as it were. We've clipped off our carbon dioxide, we've gleaned our electrons and some hydrogens to reduce NAD plus to NADH. We've hooked on our coenzyme A. Now it's called acetyl coenzyme A, and that's what will start the citric acid cycle. Now, immediately we actually get rid of the coenzyme A, and there's a 4-carbon sugar that comes up in the 11 o'clock, place on, if this were a clock, and it will attach to the leftover 2-carbon molecule here. That 6-carbon molecule will start in through the citric acid cycle. It's also known as the Krebs cycle. We're going to clip off a carbon dioxide, make waste gas. We're going to glean some hydrogens and electrons, reducing NAD plus to NADHs. We'll actually clip off another carbon dioxide. We will continue to reduce NAD pluses to NADHs. Ultimately, we will glean 3 NADHs per pyruvate or per cycle. We'll get 1 ATP out of it per pyruvate. And notice here we'll get 1 FADH2. Now, FADH2 is also an electron carrier, but it actually will start down the electron transport chain a little late, and therefore it's not worth quite as many ATPs. But just notice for now, it is an electron carrier, just like NADH, and we'll learn more about it when we get to oxidative phosphorylation. Okay, I would like to play this citric acid cycle from McGraw-Hill for you because I think it's very helpful in helping us to really understand the Krebs cycle.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle.
Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, so that is the citric acid cycle. In our next lesson, we will start oxidative phosphorylation. That is where we will finish up the process and we will harvest all of our ATP from this 3-stage cycle.